consejera y lehendakari en funciones, diputado general, rector magnífico de la Universidad del País Vasco, Euskal Herriko Unibertsitatea, científicos y científicas, señoras y señores. Es un privilegio introducir a Aaron Ciechanover, profesor del Cancer and Vascular Biology Research Center, Faculty of Medicine, Technion, Israel Institute of Technology, Haifa, Israel. Y, por supuesto, le tengo que agradecer la invitación para hacer esta presentación al profesor Echenique. La muerte, vista siempre como una tragedia, es, sin embargo, un requisito esencial de la vida. Así rezaba un reciente texto de un periodista científico refiriéndose a la apoptosis, un programa de destrucción celular definido genéticamente que permite que un organismo se libere de células que ya no le son útiles o que están dañadas o envejecidas. Como en una agrupación societaria, prevalece el interés general, la supervivencia del organismo, sobre el particular, la supervivencia de una célula. Durante cada minuto de una vida, el cuerpo humano experimenta la muerte de alrededor de 1 millón de sus células. Esta línea de pensamiento es de aplicación a las proteínas. Las proteínas mueren, cuyo recambio tiene lugar a un ritmo frenético. Al desciframiento de los mecanismos de proteólisis regulada, mediada por ubiquitina, se dedicó con fervor a Aaron Ciechanover. Su historia es la de un médico apasionado por la bioquímica. Nacido en Haifa y educado con esmero en el seno de una familia culta, media, de origen polaco, Aaron Ciechanover descubrió en edad temprana su amor por la biología, coleccionando plantas y esqueletos de animales diversos en primaria y secundaria. Por entonces, la biología era un área eminentemente descriptiva. Acababa de descubrirse el código genético y la estructura en doble hélice del DNA. Así es que inicia estudios de medicina en la Universidad Hebrea de Jerusalén y disfruta durante la etapa preclínica, ampliando conocimientos de bioquímica y farmacología, de las bases moleculares de la patología y del tratamiento de las enfermedades. Pero se desazona en el periodo clínico, constatando que el tratamiento de la enfermedad está más dirigido a la sintomatología que a las causas que la motivan. Su pasión por la bioquímica se consolida tras su graduación en medicina, dedicando su tesina de licenciatura al estudio experimental del efecto de la adición de suero en la síntesis de fosfolípidos. Cumplidos los 3 años de servicio militar, durante el cual celebra a los esponsales con Menucha, también médico, Ciechanover toma una decisión que le marcará de por vida: abandonar la práctica médica clínica y comenzar una carrera en investigación científica, fundamentalmente junto al doctor Abraham Hershko, talentoso postdoc que indagaba sobre un proceso termodinámicamente paradójico, la proteólisis intracelular dependiente de energía. Entre los años 76 y 81, sin prácticamente tener 30 años, Ciechanover y Hershko colaboran estrechamente con Irwin Rose, experto en enzimología y química de proteínas, y consiguen descifrar el modo de actuación de una proteína que resultó ser la previamente descrita como ubiquitina, cuya unión covalente a una proteína sustrato la marca como señal de muerte para que se ejecute su destrucción. Por sus rupturistas no canónicas aportaciones sobre la proteólisis regulada, Ciechanover, Hershko y Rose fueron galardonados año más tarde, en 2004, con el Premio Nobel de Química. No quiero terminar esta presentación sin mencionar unas palabras que el propio Ciechanover escribe en su autobiografía, en relación a cómo ese periodo de doctorado marcó su futuro. De mis mentores aprendí 2 principios: Primero, a seleccionar un problema biológico importante. Y segundo, a asegurarme de que contaba con las herramientas adecuadas para su abordaje experimental. Aprendí también a no ser oportunista, a ir al fondo de la cuestión, a no ser seducido por las modas, a prestar atención a pequeños detalles, a ser crítico, cabezota, a luchar contra las dificultades y a distinguir lo falso de lo verdadero y los artefactos de los hallazgos con sentido. Pero esto, dice, no lo aprendí en lecciones frontales, sino de la propia actitud de mis mentores. Pues bien, muchas gracias, profesor Ciechanover. Estos principios, en mi opinión, deberían impregnar la aproximación a la ciencia de los jóvenes y la forma de pensar de los mentores en nuestro presente. Muchas gracias, profesor Ciechanover. Suya es la palabra.
Well, good evening. Thank you, Dr. Ochoa, for this wonderful introduction. Thank you, Pedro, for inviting me to this exciting event where science is communicated to the public. I'm facing two problems, I think. One is to follow the huge shoes that Bob left behind him. And the other one is that time becomes late, but I realize that in Spain, dinners never start before 10 p.m. and people have a lot of time, but I promise you first to be light in my science, and second, to be short. I'm going to do a different job than Bob. I'm not going to tell you about my profession, which is protein degradation. I'm going to entertain you and myself with a problem that I'm sure bothers many of us, or bothers you. I'm not promising you any solution, but I think that it's really a problem that bothers also the scientific and the health biomedical community. And obviously, the dream of many people is to stay young forever. The question is, what is young? Do we want to stop growing at the age of 15, maybe 25, maybe 35? So here you see there is already a question, what is young? You know, at the Roman time, people used to live about 25 years, so being 40 was already very old. At the beginning of the previous century, of the 20th century, people lived on the average of 50 years. They died mostly of infections, so being 60 was old. In our days, we call old people that are 80, 90, maybe 100, but we are still extending our lifespan, and maybe in 100 years people live for 200 years. So you see that already the definition of being young and wanting to live longer life is very relative. It depends, when did we live? Did we live during Roman time? Did we live at the turn of the century? Are we living at the turning of the 21st century? Or are we going to live at the beginning of the 25th or 26th century? So you see, this is also type of a relative question. Then obviously the second quest is, are we going to cure all diseases? We don't want just to live longer. We want also to have a quality of life. We want to live healthier. So the question is, are we going to solve all the problems of medicine? I mean, are we going to live in an environment that in maybe 100 or 200 years or 500 years, there will be no medical schools and there will be no biomedical research anymore because we solved all problems, and then we'll focus and turn our attention to other problems. So those questions, I'm not sure that they are really bothering us, but I'm sure that each and every one of you is asking it. Each and every one of us wants to live longer again, when the word longer is very relative. And we want to cure all diseases. We want to live healthier lives that are disease-free. Now, is this realistic? I mean, are we talking realistically. And again, it's a very difficult. In science we try not to prophesize. Because you might find yourself fooling yourself very easily, even during your lifetime. But let's think historically. Let's think of Alzheimer's disease, for example. And Alzheimer's disease becomes an epidemic these days. And the autopsies tells us that if we are all going to live to 120 years, and living to 120 years is a reality because we are still extending our lifespan linearly in a rate of about 1 month every year. So people that are being born these days, they are going to live about 12 years longer than we do on the average. And I don't know for how long this curve is going to remain linear. Autopsies tells us that basically almost all people at the age of 90, when they are autopsied, they already show signs of brain degeneration that will lead eventually to Alzheimer. And if we are trying to project it into histopathological accumulation of these lesions in the brain, people predict that if we are going to live to 120 years, all of us, with no exception, are going to be Alzheimer patients. So we need to cure Alzheimer if we want to live to 120 years. We don't want all the population at the age of 120 to be Alzheimer patients. So if we think about Alzheimer's disease, at the turn of the century, during the Roman time, they didn't know anything about it. If we are reading some, medical records from the Greek and Roman times, there is no remnant to Alzheimer. Why? Because there was no Alzheimer's disease at that time, because people didn't live long enough to have the disease. Let's think about another disease, cancer. You think about cancer. Cancer is typically an age-related disease. If we are looking at prevalence, incidence of cancer, we see that it's crawling a long time, and at the 5th, 6th decade of life, it starts to deflect up, and many patients, most of the patients are, of cancer patients are at the age of 60, 70, 80, and 90. There is cancer of childhood, there is pediatric cancer, but only 1 or 2% of cancers are affecting children. So cancer is an age-related disease. Again, cancer was not a problem at the turn of the century. Very few people had cancer at the turn of the century because they didn't live long enough to get the disease. So let's say that we are going to defeat cancer, and we are going to defeat neurodegenerative diseases like Parkinson and Alzheimer, and we are going to live 150 years. Can we predict? Do we have predictive tools to tell what are going to be the diseases of the next generation? I think that not. Nobody is going to take the risk of prophesizing, and nobody is going to tell us what's behind the corner. Like we discovered neurodegenerative disease, like we discovered cancer, we may discover other diseases. So curing all diseases may be a nice quest, a nice thinking, a nice dream, but I'm not sure that it will ever happen because we really don't know what is waiting for us behind the corner. But the environment around us is much more complicated than that. We thought around 1930, 1940 that we defeated infectious diseases. There was the famous discovery of the first antibiotic by Ernst Chain and Sir Alexander Fleming. They discovered penicillin, and then came Waxman and discovered streptomycin. And we thought, wow, this was a revolution. It was truly a revolution, because the discovery of antibiotics really allowed a major breakthroughs in medicine. It allowed to anesthetize patients. It allowed to open them in an aggressive way, to walk into the operating theater. And now we are doing it in a much minor way, as Bob mentioned to you. But it really revolutionized the way that we are living, also understanding infections, sanitation, sterility, a sewing system, living in a much cleaner environment, really revolutionized our life and allowed us, this is one of the major factors that allows us to live longer. But we see that new infections are coming. AIDS is a new infection. It was discovered only in the early 80s. We didn't see AIDS before. Ebola, SARS, avian influenza. All those are new diseases. Luckily, we are able either to contain them or that the agent is sensitive to the environment, like the AIDS virus, and we learned by behavior to avoid it. But nobody guarantees us that the infectious diseases will not come back, and in a stronger way, and they do come back. in a stronger way. We are seeing now much more aggressive forms of tuberculosis that we thought is out. So even diseases that were defeated are coming these days in new and more dangerous forms. So I'm not sure that the quest to end illnesses, to end diseases, is a realistic one, and all what we have to do is work hard and solve our current problems. We shouldn't be desperate, obviously, and we shouldn't stop working. But I think that this is maybe one way, if we believe in God, in any God, whatever God is, of balance inside nature. I mean, diseases are signs of degenerations, are signs of age, of long time, and probably living organisms are not built to live forever. Like materials are not built to live, to stay forever, and they are undergoing changes. But that's going more into philosophy. But I think that it will be very interesting now to come down from the 30,000 feet that I'm trying to fly with you on these very interesting philosophical questions. And, this is another one. Bob, this is taken from Judah Folkman from the Time magazine, with his thought that we are going to put an end to cancer by curbing the blood supply to tumors, and it turned out to be just one approach, because tumors are very sophisticated. These cells can, if we close the door, they will come from the window. If we close the window, they will come from the electric outlets, and this disease turned out to be a very complex disease. But again, this was again part of the naive dream of the late 70s and maybe early 80s that, we found one solution to the disease, which tells us that we are not going to wake one day up and find one magic bottle with one drug that will end this disease and so for many other diseases. But there is a way to fight diseases. And if we look at diseases in a big way, we shall find that many of the diseases are man-made, and therefore we can prevent them. It's much cheaper and much more logical to prevent diseases than to take a patient to the hospital and to treat the disease. So think about this disease, which is an epidemic in the Western world, obesity, metabolic syndromes. This disease, so-called, can be prevented. We just have to control it. And each and every one of us has to control it. But if you think about obesity, and the Lasker Award last week went to people who really dissected obesity on a molecular way. They found the leptin and the leptin receptor. I'm not going to burden you and to bother you with all the details of the biochemical mechanisms of obesity. But we are going to treat obesity, obviously, with drugs. But obesity in many ways is a man-made disease. We hurt ourselves. Look at this disease, smoking. I mean, this is kind of an exaggeration of somebody who smokes 200 cigarettes at one shot. But, think of lung cancer. Lung cancer is mostly, and many other damages are caused by smoking. And tobacco is something that we are growing, we are fostering, we are nurturing, and then we are manufacturing and we are smoking. These diseases that we are also contaminating the environment can be all eradicated. So this is one way to really cure part of the diseases. Think about environment. We are polluting our environment, and now we are fighting back. We are trying to be green. We are trying to curb air pollution coming from cars, from the industry. But again, we caused all of it. The Industrial Revolution brought it all on us, and now we are trying to take steps back and to prevent it. Genetic diseases, familial diseases, that's more sophisticated, but I'll come back to it. I think that by better screening and manipulations, we shall be able also to prevent, or at least to predict, genetic diseases. And I talked about infectious diseases. Infectious diseases are partially environmental diseases, and they can also partly, like we talked about AIDS, can be prevented by understanding the pathogenetic mechanisms of these agents. So you see that many, many diseases can be controlled by simple measures. We don't need to develop very sophisticated operating theaters and delivery and imaging devices and delivery devices. We just have to behave better and mostly to take good care of our environment. And this will solve many of the problems. And I think that many countries and organizations, and this must be done on a global way, on a global basis. It cannot be that Spain or Israel or one single country will take care of the environment and the next neighbor and the next neighbor country will continue to pollute the rivers and the air that then will flow to the other country. This must be done on a global way, and I think that creating organizations like the EU is one way to respond to such huge challenges of handling the environment. But let's go now even further down and try to see what really medicine is preparing for us in the 21st century. And I'll be very brief, and I divided the drug development 20th century into 3 periods. And I think that it's right also to think that the 20th century was the century of drug development. There were many drugs beforehand, but doing systematic clinical experiments, having 10,000 control patients and 10,000 experimental patients, working out the statistics of the drug effects, comparing different drugs, is something the big drug companies, Merck, Novartis, Johnson & Johnson, all these companies are all creatures of the 20th century. Until the 20th century, everything was very erratic. People relied more on herbs, on chemistry was not well developed to purify ingredients to understand mechanisms. So we are basically talking the 20th century. And in the 20th century, it will be very interesting to follow what happened and therefore where we are going to head just in the next few years. So we are entering the third revolution, but in order to understand the third revolution, I think that it's better to go to the first revolution. And the first revolution, I don't call it revolution, it was the era of incidental discoveries. We are talking about the 20s and the 30s, and always it's very helpful to bring examples. And one example is an unbelievable efficient drug, which is called aspirin. I'm sure that if I'll ask people, Bob tried it, let's don't shy out. How many people in the audience are taking aspirin? Me? Well, quite a number of them. So aspirin is really a magic drug. It's not a recommendation from a physician for you to take it. It has side effects, gastrointestinal bleeding, and so on and so forth. But aspirin turned to be really a magic drug. It prevents mostly coagulation, blood coagulation, aggregation of platelets, and it's recommended mostly for people after heart attack, but people claim, though the New England Journal of Medicine came with a recent paper showing that it's wrong, and I still believe that it's right, but it's only a belief, that it can be used also prophylactically to prevent heart attack. I luckily didn't have any heart attack, and I decided many years ago, along with many of my physicians' colleagues, to take it, and at least it didn't cause any damage. But aspirin started from old Egyptian literature when they discovered something that in the willow barks that is very bitter, and it took away pain. So it was discovered initially by the old Egyptian, but then people forgot about it. Then it went through some French scientists who purified the material, which was salicylic acid, which was still very bitter, and then they made it less bitter by modifying it. But it was rediscovered by a Jewish scientist by the name of Felix Hoffman that worked in Bayer. And Felix Hoffman's father had a disease that is called rheumatoid arthritis. And rheumatoid arthritis is a devastating, debilitating disease in which the joints are inflamed, both the foot and the hand joints are inflamed, and the people are really suffering, and they are severely handicapped. And actually he wanted to remove pain, to treat his father with a pain relieving agent, and then all of a sudden he found that the drug has an unbelievable anti-inflammatory properties, and it was later discovered that the drug prevents the synthesis of inflammatory mediators and so on and so forth, but I don't want to go into the mechanisms. But after Felix Hoffman tried the drug on his father, he went back to the company and suggested that the company will manufacture the drug as an anti-pain, anti-fever, and anti-inflammatory drug. Bayer accepted it, and they start to manufacture it. Needless to say that Hoffman didn't see any penny out of it. Actually, because he was Jewish, he was fired from the company later on, so he was even punished for the $1 million, for the many millions of dollars that the company made. And aspirin is a very simple chemical. Every student can make it in the basement, and it's probably the best sold drug ever. And it was rediscovered as a drug that can prevent aggregation of platelets and probably prevent a second heart attack and so on and so forth. You can buy it hundreds and hundreds of grams for a few dollars in every pharmaceutical chain. And it's probably the drug that is consumed in tons, still in tons, in the world, and it will continue to be consumed in tons. People claim that it can also prevent Alzheimer's disease and neurodegenerative disease. We still haven't discovered probably even the tip of the iceberg of this drug. It's all serendipity. Felix Hoffman, his brother, there was no science behind it. It was just him reading the literature and going on. Let's go to penicillin that I just mentioned. Again, I'm not going to tell you the whole story. It's complicated. You can read it, but some of the basic discoveries was an experiment made by Alexander Fleming. He was a microbiologist, and here you can see the bacteria. He left the Petri dish on which bacteria grow on the table. He left it open, and for a microbiologist this was a mistake, but during the time that it was left open, a fungus landed on this rich material, and the fungus grew. It was Penicillium colony. It was a Penicillium fungus, and Alexander Fleming noted that there is a halo around this colony in which bacteria didn't grow. So he surmised that the fungi probably secrete an antimicrobial agent, an antibiotic. Bios is life, anti is anti, something that prevents the proliferation of bacteria. And then Ernst Chains, before and after there were indications for it, synthesized and purified the material and then synthesized it chemically, and penicillin came to the world, and we entered a new era in medicine that I just mentioned. Again, serendipity, good eye of a scientist. So there was no science behind it. At that time, there was no science. Let's enter the second revolution. And the second revolution, I think, reflects chemistry. It reflects the strengths of chemistry in the 50s, 60s, and 70s, when people gained huge synthetic ability. People were able to synthesize basically every compound. I mean, to analyze it via all kinds of wonderful devices, and to understand the very delicate structure, and to synthesize libraries of millions of compounds. So this I call the second revolution. We are still in the second revolution, and the second revolution is a revolution of what we call brute force, screening. You have cancer, you take cancer cells, you grow them on small dishes, millions of small dishes, and you try compounds one by one. So you have to dissolve them, and there are many, and you are looking for leads. So it's a random search. You have no idea about the mechanism, but you assume that among the millions of compounds that are there in the libraries, and you can buy the libraries from companies, 1 or 2 or 10 or 20 or 100 will be found as efficient anti-cancer drugs. And you can develop many models, not only to cancer, but to other diseases, and you start a screen. It's a brute force screen. From the screen, 100 compounds will come. You take these compounds and you transfer them further into animal experiments and then into safety experiments in human. You're going to phase 1 and into phase 2 and into phase 3. Very few, if at all, will make it to become a drug because of many reasons. I will not go into it. But at the end, those compounds that will make it, or many compounds that made it already to the market nowadays, are compounds that started as leads in screens, and then they were modified and changed further by chemists in order to make them more soluble, less toxic, and so on and so forth. But basically, if we look at this approach, it's, in my opinion, and it should be continued, obviously, a non-intelligent, quote by quote, brute force approach. We just screen. And I'll give you just one example of an awfully successful drug that I'm sure that if I'll ask you to raise hands, many more hands will come up. And these are statins. Statins are anti-cholesterol drugs. They are used to reduce cholesterol. This is a magic drug. And unlike aspirin that is sold for few dollars, several hundred grams, This is still sold for a lot of money. It's still under several patents. And the market for statins last year was about $25 billion. $25 billion, the market for statins. This is a magic drug. It lowers cholesterol, and by this prevents further aggravation of occlusion of the coronary arteries and heart diseases and other vascular diseases, accumulation of cholesterol. And it was discovered by a Japanese pharmacologist, Akira Endo, that won for this the Lasker Award several years ago for medical research. And this came out of a screen. He looked for anti-cholesterol drug, but he didn't look for the disease. He looked because he thought that cholesterol is part of the wall of parasites. Actually, by preventing the synthesis of cholesterol, parasites won't be able to survive. They won't be able to synthesize the wall. So he looked for parasites and for anti-parasite drugs, and then these statins, the first statin came. Now we have better statins. Probably you know some of them. The most modern versions are called Lipitor or Atorvastatin. I'm sure that many of you know the names, and it became a blockbuster. And this blockbuster, and there are many of them. I can just keep on running and tell you about the success of chemistry as a source of novel drugs. We are exiting this era. I'm sure that it will take us several more years. Again, I don't want to prophesize how many decades or how many years we are still going to spend on screening, but we are clearly entering the third revolution. And this is the era of design and fitting. So we are out of brute force screening, and we are going in the language of Lee Hood, again, one of the giants of biotechnology and mass analysis of processes, what he called the medicine of the 4 Ps. One, it's going to be personalized. We are going to fit drugs to the repertoire, to the specific disease of the patient. It's going to be predictive. We are going to be able to predict diseases, and I'll come back into it. Because it's going to be predictive, it's going also to be preventive. Because if we shall be able to predict that somebody is going to have diabetes or heart attack in 10 or 20 years, we are going to do all what we can in order to prevent it, to fit a diet, to treat with preventive drugs, prophylactic drugs, and so on. But most importantly, or as importantly, it's going to be participatory. We are going to participate, to involve the patient in the process, because we are going to penetrate deep into a very sensitive information that the patient owns even without knowing. And I'm going to come into it shortly. So Lee Hood really abbreviated this type of approach as the 4 Ps approach: personalized, predictive, preventive, and participatory. And I'm going to stop on each of them very briefly. But what really caused this recent development, which we are into for, I would say, the last maybe 10 to 15 years, even without knowing, but we are moving fast into it. It's mostly the Human Genome Project, and we are celebrating 10 years to the unraveling of the human genome. Actually, the human genome was unraveled the Nature paper came out in April of 2000. And the human genome is the entire information embedded in our DNA. We were able to read the genome, the entire DNA, and its information. Reading doesn't mean that we understand. Reading doesn't mean that we can predict. Reading doesn't mean that we know what it means, but we are able to read. It's like a collection of letters. It means, I can read Spanish because I know the Latin letters, but it doesn't mean that I understand Spanish. It doesn't mean that if I read a line, I will be able to understand what's written there. So that's what we did, and the future will enable us, hopefully, also to understand what we are reading. But the whole library is there in front of us. So it's the library of the DNA, and you can see the Time magazine, and I'm going just to Popular Science, already predicted that the drugs of the futures are going to be based on our DNA and on the information. But things got much more complicated. Now, what's wrong with the current medicine? What's wrong with screening? Screening, and what's wrong with the current medicine that we have? Well, screening, as I told you, starts with cells, cancer cells, or whatever model we have to the disease, and we are making a lead, and from the lead we are going on to develop. But this screening assumes that cancer is one disease. Let's say take breast cancer. That breast cancer is one disease. Woman walks into the clinic and she tells the doctor that she felt a lump in the breast, and the doctor looks into it and he's using a very sophisticated imaging techniques. He's using MRI, initially CAT scan or MRI, and he's using a biopsy and whatever, and he looks for metastasis and so on and so forth. But lo and behold, when the woman asks the doctor, doctor, tell me, am I going to make it through? Am I going to survive the disease? The doctor tells her, well, it depends on the disease. 50% you have chance to survive. 20%. If you have a brain tumor that was mentioned by Bob, the glioblastoma multiforme, probably the doctor will tell the patient, if he has the guts, you have 0% these days to cross 5 years, or less than that, much less than that, or pancreatic cancer. But in breast, let's say the doctor will tell her 50%. Then the woman asks the doctor, what do you mean, doctor, by 50%? The doctor tells her, you know, I'm a very experienced doctor. I'm 70 years old, and I've seen 1,000 women like you. Exactly. And all I can tell you is that 500 died and 500 made it. That's all I can tell you. It's based on statistics, of similar cases statistics. But I cannot tell you more. And when the woman asks him, doctor, how I'm going to know, he tells her, you'll have to wait for 5 years and then we'll see. And obviously, I'm oversimplifying the story, but the 5 years are accompanied by repeated examinations, coming back to the clinic, seeing if the disease didn't spread, and so on and so forth. Scientists didn't like this solution of statistics. Scientists wanted to understand what is the difference between the two women that walked into the clinic of the doctor. The one that made it healthy after 5 years, and the one that suffered severely and at the end of the 5 years died. What is the difference? Though initially they presented with apparently the same disease. If we want to reword, to rephrase the situation, actually, this woman did not have the same disease. They had the same lump, they had the breast cancer, but we didn't have, the physicians and the scientists, we didn't have spectacles sensitive enough to tell the difference between the two. Because apparently they had the different disease. We treated them the same. We took the lump out by surgery, we treated them with chemo, we irradiated them, but the fate of the two women was completely different. And the same is true for men that walk with prostate cancer, or with a man or a woman that walk with a stomach cancer, or with a colorectal carcinoma, doesn't matter. All those diseases that are not defined as completely fatal diseases like brain tumor, like glioblastoma multiforme. So we say, well, the tumor is isolated and it didn't send metastasis. Again, what do we mean by it didn't send metastasis? It didn't send metastasis by the criteria and by the threshold of the test that we have in order to find metastasis. But how do we know that one single cell didn't settle up in the bone or in the brain or in the lung of the patient? We don't have sensitive enough tools to tell whether one cell, one single cell, was already sent and is going to develop into tumor. In half a year, when the patient will come back, we may be able to say, but that may be too late. When a bone metastasis is already developing, it's, well, too late to help the patient, certainly for lung metastasis. So the diseases are different. And we failed until now, until these days, to classify the diseases, and in the language of a wonderful scientist, Mary Claire King, from Seattle, breast cancer is not a disease. Breast cancer is a collection of different diseases, small groups of different diseases. And the same for prostate cancer, and the same for colorectal cancer, and the same probably to most of the diseases. We just, in our naivete, and in the primitive tools that we are equipped with, classify those diseases under the same umbrella. But they're absolutely not the same disease. Because the fate of the patient is the only factor that tells us that these are not the same diseases. If one patient dies and one patient is living happily, you can tell that the diseases are very different and in a vast major way. So the challenge of science is to find the differences. And the difference is embedded in the molecules, in the understanding of the molecules. And again, you can see we start with a group of patients that we don't see the colors, unfortunately. So there are the green patients and the blue patients and the yellow patients. So this is a group of breast cancer patients or prostate cancer patients at the day of the diagnosis. 5 years elapsed. Few of them didn't benefit from the treatment and died. And some of them, or many of them, did benefit from the treatment. So it's only after 5 years that we are able to identify the different colors. At the beginning, we are blind to the colors. And we want not to be blind to the colors at the very beginning. We want to predict, we want to know. We don't want only to know who is going to die. We want to know why he or she are going to die and to develop drugs that will fit this specific group and benefit them. So it's not only the matter of curiosity, of prediction, it's the matter of predicting initially in order to develop the proper drugs. So this is a major challenge to subclassify those diseases that are still hidden from our insensitive tools of diagnosis. And here again, it's coming again and again in popular journals. And I just want to show you one example that we are using today. We have 2 biopsies, a needle biopsy that was taken from a breast cancer. And this woman doesn't have the brown staining. It's a histochemistry, it's just a staining. You can see that this is woman B and this is woman A, and woman B has something that woman A doesn't have. She has an estrogen receptor. And therefore, and the estrogen receptor was shown to be one major causative agent, this mutated estrogen receptor in the disease. So therefore, we can fight the disease of this woman with tamoxifen, which is an anti-estrogen receptor. But it will be useless to treat this woman with tamoxifen. Why to treat her with tamoxifen if she doesn't have a mutated estrogen receptor? And the same for Herceptin. Herceptin is treating women with breast cancer that has a mutated EGF receptor. But women that have a normal EGF receptor, it will be not only waste of money, but mostly dangerous for the woman. We are treating her with a useless treatment instead of treating her with something that will be useful for her. So it even impedes our progress. So it's not only waste of money and waste of hopes and implanting unnecessary hopes in these patients, but it's really doing very bad science and very bad medicine. It's malpractice. So this is what we call molecular medicine. And the purpose of modern medicine is to expand it. Now, this estrogen receptor was discovered by serendipity or by curiosity of a very talented researcher, and there are few of them, but we want to get the full map. We want to have the full road map of our repertoire. And the road to it goes via, and this is Herceptin. Again, breast cancer, you need to have a mutated EGF receptor, and only then Herceptin will be helpful. Otherwise, it's useless. So this is called personalized medicine. By personalized, I don't mean that every one of us will be identified by the Israeli government or the Spanish government according to the ID card, and a bag with the drugs will be prepared for us, and if we are sick, we are calling the phone, and the bag will be sent directly to our mailbox. This is not what we mean by personalized medicine. Personalized medicine means that we are fitting the drugs that are available, but mostly those that will be available, to the specific repertoire and the pathology that affects the patient. That's what we mean by personalized medicine. It doesn't mean that 7 billion people that are walking on the face of earth, each and every one of them will have its own specific drug waiting for him or her in a bag. That's not the meaning. But the meaning is personal fitting after the diagnosis and according to the repertoire. And the road goes via the human genome that I told you, understanding the entire genetic repertoire. It got more complicated than that because initially, again, science, Mother Nature is very hard to crack. Whenever we crack a problem, 2, 6, 10 more are opened, and actually was a wonderful statement. It was a short speech given by David Gross, who won the Nobel Prize in Physics in 2004. And during the banquet dinner, he asked whether there will be Nobel Prizes in 10 years or 100 years or 1,000 years. And the question came from the notion is, will there be a day that we shall know everything? And at that day, there will be no new discoveries and new inventions, so there will be no Nobel Prizes because there will be no new discoveries. So we shall dismiss the Nobel committees and send them all home, healthy and living forever. Well, his prediction was, and I tend to believe in it, and again, we are here flying back high into philosophy, that knowledge is not only endless, but it's endlessly expanding, infinitely expanding. And I think that he expressed it in an unbelievable literal way. That the single most prominent and clear and obvious product of knowledge is more ignorance. We are generating knowing more, we are generating more ignorance. So, we were very naive 20 years ago and 10 years ago when the genome was discovered that that's it. This will be the solution for all our troubles. And again, but then it turned out to be much more complicated because the genome itself is not stable and it's undergoing changes. And then the products of the genome, the RNA and the proteins, and I promise not to bother you with chemistry and with the central dogma of biology, are also dynamic and they undergo post-transcriptional and post-translational changes. And it turned out to be much more complicated. But nevertheless, this is the road that we have to go through. There is no other way. We need to cross and to traverse this way to understand initially the mutations in the DNA, and then to sort out the chickens from the eggs, and then to develop drugs to whether it's the chicken or the egg, it depends how one looks at it, and so on and so forth. And a new project was launched in the United States, but now it's a worldwide effort, and that's sequencing numerous genes. You'll ask me, well, you just told us a few minutes ago that the genome was sequenced. Yes, the genome of one single person. And the genome of one single person took almost 10 years to sequence and cost hundreds of millions of dollars. But with new technologies, we are approaching a time that is just behind the corner, a few years from us, in which we shall be able to reduce the sequencing of the genome of each and every one of us to less than $1,000 and to be carried out in few hours. People even predict that in less than few hours. So it's going to be like a typical sophisticated lab examination, like MRI, that costs still a lot, but available to everybody in the developed world. So we are going to be able to sequence the genome of each and every one of us, but we already started. And we started mostly at the Broad Institute, but it became the MIT run by Eric Lander, but it became a worldwide effort, and countries are sharing the task, and people decided that from each disease, they are going to sequence 500 genomes. This will be sufficient to cover most of the mutations. I don't want to go into the analysis of why the number 500 was. So we take brain tumors and pancreatic tumor and liver tumor and gallbladder tumor and muscle tumors and whatever tumors, and then we take other diseases, inflammatory diseases and neurodegenerative diseases, and we are sequencing them. We take samples from the patients and we are sequencing them. And once we'll have 500 made, we shall be able to compare them. And we shall be able to tell what is the difference between the diseased tissue and the healthy tissue. Obviously, we shall need lots of bioinformatics and filtering information. We shall have to filter lots of noise and what are driver mutations and what are bystander mutations. There will be mutations that are causative and there will be mutations that are hitchhikers that do not belong to the story. But again, I'm not going to bother you. Once having all this information, we did quite a long road forward. We shall be able to pull out these genes and then to look into the products and to see what happens to the products, to the mutated products. And using animal models and again screening, we shall be able to identify new targets that we still don't know. The idea is to identify targets, but the target will be specific to the patients, because now when a cancer patient will walk into the clinic, the first thing that he will do, he will give a blood sample or a buccal smear sample to the doctor. The doctor will sequence the DNA and the doctor will know to which group of cancers he belongs, and hopefully, again, I'm prophesizing, we are still decades away from this solution, we'll be able to fit the proper treatment to the proper disease of the patient. Now, let me just finish with a word of caution. I try to be optimistic because I see the road, and it's not my road, it's the road that is being paved by many, many scientists and many, many countries, and it's a multinational effort. But the road is not devoid of obstacles, and not only scientific obstacles. I talked about the scientific obstacles, and I hope that people will be able to overcome them. For sure, some of them we shall be able to overcome, but they are bioethical, and we have to think about bioethics. We shouldn't forget that at the end of the day, it's the human being that we are going to treat. It's not a microbe, it's not a virus, it's not an object, it's a human being. And human beings are sensitive creatures, and they are very complex creatures. They are coming from different backgrounds, from different faiths, from different cultural backgrounds, nations, languages. It's a most fantastic phenomenon to live in this world and to see this diversity of the fish in the aquarium, but on the other hand, it poses huge problems. Now, once having the DNA of every one of you, or people that will volunteer to give the sample, becomes very dangerous. Because though now we cannot read it, in few years or decades we shall be able to read it. And then we shall have at our hands a very sensitive information. We'll have an information that is predictive. We shall be able to predict the diseases that you are going to have. And people may not want to know about the diseases that they are going to have, and they may not want that others will know about it, because for time being, for now, every piece of information that is related to our health is held confidentially by the doctor, and we trust the doctor. And one of the major statements in the swear, when the doctors are being sworn into the profession, still the Hippocratic swear, but also more modern version of it, that the doctors should never disclose the secrets of the patients. And this is kind of a moral contract that we have. And here the doctor is going to have in his hands a much more explosive information that we can ever imagine. Much more explosive. About our future, maybe mental states and certainly physical state and so on and so forth. But let's say that we trust the doctor. Everything will work fine. The doctor will put the information in a very safe. And it will be 30 centimeters thick steel, and it will be buried 20 meters below ground, and it will be with alarm system. Nobody will have access to it. The question is whether we want to have access to it. Let's say that a woman has a gene that increases by 80% her sensitivity to breast cancer. And there is such a gene that is called BRCA1. So the woman has the gene, and she got breast cancer, and the breasts were taken out. Now the question for the woman, is she going to tell her daughter about a potential risk? Because the daughter may have a chance to have the same gene. And the daughter is still not sick. So this, we are already, you see, we are walking on a very thin grounds now. Well, maybe the mother would like to say, no, nobody is going to know about it. But then the mother faces a moral question. Am I going to tell it to my daughter? And if I'm going to tell it to my daughter, who is now 20 years old and she just graduated second year college, is the daughter going to tell it to her boyfriend that she is going to marry? That she has a huge risk of having breast cancer, and she decided to do, let's say, two-side preventive prophylactic mastectomy. I mean, we can think of all kinds of potential. So here we are walking away from the legal system. Let's forget about law, let's forget about parliaments. Let's think about human beings, how they behave in face of such an information. What they are going to do with such an information. And BRCA1, that is already known, is just one example. We are going to have hundreds of those. And what if couples are going to be married, and already now, people are used to have prenuptial agreements. They look into the economy, they have all kinds of agreements. People would like to know what is the health state. Obviously, if I'm going to marry now, and I have only one eye, well, some people will not like the idea. Some women will not like the idea that I have only one eye, and they will not marry me. And you know that for handicapped people, for overt handicap, it's a major problem to integrate into society. But having a mutated gene that renders you susceptible to cancer in 10 years is also a handicap. We still don't know it. It's very hard for us to grasp. But think about it. It's a handicap. And the information will be behind the corner. What we are going to do with this information. Maybe our fiancé would like to know about it. Or maybe we would like to know it about our fiancé. So this is not science fiction. Well, for now it looks like a science fiction, but we are approaching it. So as I told you, you're not going to walk out of this hall with a solution. You're not going to walk out smiling that during your generation people, scientists in the laboratories are going to find cure to all diseases and to keep you young forever at the age of 25. All I wanted to pose for you are the bioethical and the other questions that scientists are facing, but the bioethical ones are not for scientists to solve. This will have to be solved on a very broad societal base, involving clergymen, sociologists, psychologists, and so on and so forth. But it's coming. Science is not going to stop. The human genome is there. People doubted that scientists will ever be able to do it, but they did it in even shorter period than was predicted. People thought, well, it's millions of dollars, it's only one man. No, within 10 years we are able to do it on each and every one of us with less than $1,000 and less than few hours, and it will be even cheaper and even shorter time, and we are progressing. But side by side there are problems, scientific as well as moral, and as a society we ought to solve them. So all I wanted is not to answer questions, but rather to raise them up, and to thank again the organizers, not only for inviting me, but for a most important mission that they are carrying out, and that's communicating science to the public. I think that this is also a moral obligation of scientists. We are using the taxpayers' money, and we cannot use the argument, trust us, we are the smarties, we shall solve the problem. You will keep on paying taxes, we shall build nice institutions and nice research universities and solve your problems. It's our duty to communicate the happy minutes as well as the unhappy ones, as well as the problems, to the society, and to discuss together with the society the future of where science is going, and mostly the future of the effects and the impacts of science on society. Thank you very much, and wonderful success in this meeting.